Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last class, we have started discussing differential gene expression analysis or DGE analysis in short. We have talked about the preliminary concepts and we have also started discussion on DSEC2, right. So, we have completed some parts, right, about the steps that we need to do, right, for DSEC2 analysis. Okay. So, we will continue that discussion in today's class. We will look at the results that are generated by this DSEC2 tool and also will visualize the results, right. So, once we generate the results, this is a huge results and we need to use some visualizations to actually better understand what we have got. Okay. So, let's the agenda for today's class. So, we will look at the DGE analysis results, we will generate those results and we will look at some of the visualizations and some of the plots that are generated for visualizing the results. Okay, so, just to briefly summarize what we have discussed in the last class regarding DSEC2. So, DSEC2 starts with raw count data matrix. It utilizes normalization method with median of ratios method, which can take care of some of the technical variations in the data. And then it uses a model that is based on negative binomial distribution, right. So, this is a parametric method, it has some assumption about the count data distribution and it models them using this negative binomial distribution and then it fits a generalized linear model, right, and it takes that approach to estimate the parameters, the coefficients that are fit in the model. Okay. So, those coefficients give us the mean expression values of the genes as well as the differential expression, right. So, whether some genes are showing a difference in expression, right. So, in terms of differential expression we get the log fold change value, okay. So, or LFC. All right. So, again I will remind you the terms, right. So, we start with count matrix with count Kij for gene I in sample j and DSEC2 assumes that Kij follows negative binomial distribution with mean μij and dispersion alpha I. The steps that are remaining, right, the first step is we want to estimate the dispersion alpha I, right. So, once we have estimated the parameters beta from the model, right, we need to identify, okay, whether this beta is significantly different from 0, right. So, that will tell us whether a gene is differentially expressed or not. Okay. So, to do any statistical test we need to also get an estimate for this dispersion alpha I. Now, this is quite challenging, right. We have to use the data itself to estimate this dispersion. Okay. So, what is this dispersion? So, this is actually a very critical parameter of the model as you now understand. For doing the statistical test we have to use this parameter, right, because we are assuming negative binomial distribution and this dispersion alpha I is a parameter of the distribution, right. This will determine the shape of the distribution. So, alpha is actually related to variance of Kij using this term, right. So, variance of Kij is given by mu ij plus alpha I mu ij square, right, and this models the variability within group and between replicates. Okay. So, we can estimate this alpha I, right, from the replicates that are present in our data, but as I mentioned in the last class, the number of replicates is very small, right. So, it is usually 3 to 5. Okay. So, it is a very small number of replicates because we cannot do these experiments in large number of replicates, it is difficult, they are expensive, right. So, we would have to settle for only 3 to 5 replicates, and which means the estimates of this dispersion or alpha I could be noisy because we are estimating from only a small number of replicates, right, because maybe one replicate is slightly out, like showing slightly different pattern in expression and then of course, the alpha I would be noisy. Okay. So, how do we address that, right, how do you address that issue? Okay. So, it is actually addressed by something called dispersion shrinkage. Okay. So, we want to reduce this noise in the dispersion data and the idea is very simple and there is an assumption. The assumption is that genes with similar mean expression should have similar dispersion value. Okay. So, this is an assumption that we make, this model DSEC2 makes, right, and based on that it applies an empirical based framework to shrink dispersion towards this expected dispersion value, right. So, if you look at the original reference, you will see, right, that once we calculate this difference this dispersion value for each gene. For some gene the dispersion will be much higher than the group of genes that show similar mean expression, right. So, there is a relationship between dispersion and mean expression. Okay. So, what this model does, right, it assumes okay, genes that have similar mean expression also should have similar dispersion value, okay, because we are talking about within replicate measurements, right. So, there is no reason why the dispersion value should be different. Okay. And then it uses this framework to actually shrink this dispersion towards this expected dispersion value, and this shrinkage is actually dependent on the number of replicates. Okay. So, if there are higher number of replicates, it means there will be less shrinkage, right. So, because if you have more and more replicates, you are more confident about the dispersion estimate. Okay. But if you have very small number of replicates, maybe you have only 3, and then in that case you may not be that confident about this dispersion estimate. So, which means you need to have you need to probably shrink the dispersion more. Okay. So, this is kind of the assumption behind this dispersion shrinkage. Now, once we have estimated this dispersion, right, so we also calculate the log-fold change, right, and there is another term that we also use is called log-fold change shrinkage. Okay. What does it mean actually? This is also somewhat related. So, what we see in RNA-seq data or also in microarray data is that genes that have low read count or low mean expression, they actually show a lot of variability in the data. Okay. The technical replicates, they show a lot of variability, right. This is kind of expected, right. If you have very small number of reads to choose from or small number of fragments to choose from, right, there is a lot of variation, right. So, if you are doing the same experiment, so you can imagine this as a pool of molecules, right, that you have, and then you have, for these genes you have only a very small number of molecules. Now, from this pool, for each technical replicate you can choose samples, right. So, you are sampling from that pool, and the genes that are underrepresented, right, or have low expression, there is likely to be a higher variation between these replicates, right. So, when you are pulling when you are sampling this, right, for these genes, in some replicate you might get 10 reads, in some replicates you might get 5 reads, in other replicates you might get 2 reads, right. So, this is what we see, we see higher variability in genes with low read count. Okay. And to account for that, again we want to shrink this log-fold change value, and again DSEC2 uses an empirical based framework, right, and of course you can use different frameworks to do this shrinkage, and this actually gives more robust results. This has been seen by the researchers. Okay, so the next part, now we have estimated the dispersion, right, we have fit the model, we have the parameter values estimated using generalized linear model, right. So, we know the beta values. Now, it is time for the hypothesis testing, right. So, in the hypothesis testing, in the simple example that I gave you in the last class, we have null hypothesis, right, which says okay, beta values are 0, which means there is no difference between disease and healthy condition. Okay. So, by hypothesis testing we actually have this statistical basis for that, right, by saying okay, whether we can reject that hypothesis or not, right. So, this is what DSEC2 does, right. So, precisely we are testing whether coefficients are significantly different from 0, right, and coefficients that are significantly different from 0 and those genes will be identified as differentially expressed gene. So, to do that, DSEC2 what DSEC2 does is it actually will divide these log-fold changes by the standard error, and it will test against a standard normal distribution using world test. Okay. So, you will get this world test statistics and the p values after the hypothesis testing for each gene. Okay. And these p values will be their significance that will signify, right, whether we can reject the null hypothesis or not. Okay. And I have also mentioned this because we are doing this for a large number of genes, right. So, sometimes 10,000, 20,000 genes, we have to have something called multiple hypothesis testing, right, because we are doing this hypothesis testing for each gene, right. So, individually we are doing 10,000 maybe hypothesis tests, or if you have 10,000 genes or 20,000 hypothesis tests. So, we need to do something called multiple hypothesis testing. Okay. We will talk about this multiple hypothesis testing in the next class, right. So, this is something very important for getting accurate results. Okay, so I will very briefly mention about the other tool, which is the AJAR tool. Okay. Here is the reference, and this is also a bioconductor package, and again this is a very popular tool for differential gene expression analysis. So, AJAR actually uses very similar concepts, similar steps, but there are some differences. Okay. So, AJAR also starts with raw count data, and it does the TMM normalization to generate the normalized count data. We have talked about TMM normalization, and again we have seen this method can account for some of the RNA composition bias that might be present, right. So, this is the normalization that is used by AJAR. So, AJAR is also a parametric model, right. So, it uses the negative binomial distribution. So, read counts are modeled using this negative binomial distribution, and it also uses a conditional maximum likelihood approach to estimate gene wise dispersion values, right. So, you see the similarity with the other tool TSEC2, right, but of course, there are some differences. If you go into the reference and see the details, you will see the dispersion methods are not exactly same, right. So, this estimation process and the shrinkage of dispersion, again it uses empirical based method, but there is some difference. And once one of the unique points about AJAR that it can actually sometimes separate out this technical variation with biological variation. So, it can estimate the biological variation while calculating dispersion. Again there is some underlying assumption. So, it assumes the count data within technical replicates, it follows Poisson distribution, whereas between biological replicates you have the negative binomial distribution. So, it can once with that assumption it can actually calculate the biological variation while calculating dispersion. And for statistical testing or hypothesis testing, right, we have seen for DSEC2 it uses wild test, but AJAR uses Fisher's exact test for differential expression. Okay. So, to identify these differential expressed genes, it will use something called Fisher's exact test. So, what is the advantage of Fisher's exact test? Because it can work with very small number, it is an exact test. So, it can work with very small number of data points. So, even if you are working with only 2 replicates or 3 replicates, it will give you more robust results, okay, because this is an exact test that is used. Okay, so, we have now talked about these two major tools, two most popular tools for doing differential expression analysis. What I am going to do now is I will show you some results by the analysis using DSEC2, right. So, again I will take the results from the 16 samples that I am using for all these illustrations. So, we have raw count data for 16 samples and divided into 4 groups. So, each group has 4 replicate measurements, right, and this is how the data looks like. This is the raw count data, right. So, what you can see here, right, on the left column, right, the leftmost column is you have the gene names, right. So, these are the genes, right. So, these are the genes here, and here are the samples, right. So, starting from S1, S5, S9, okay, and up to there will be total 16. Of course, I cannot show you the whole thing, right, all the columns here in this small space, but you have 16 samples, okay, and for each sample these are the count data, right. So, you can see for gene ASC1 in sample 1 you have 543 reads mapping to this gene, okay. So, these are ordered in such a way that these replicates are side by side. So, S1, S5, S9, S13 these are replicates of each other, okay. So, this is how these are organized, okay. So, again you have list of 7,000 genes, 6,000, 7,000 genes for which you have this count data, okay. And as you can probably see, right, the count data varies a lot, right. So, for some genes you have very high count data, for some genes you have very low count data, right. So, you have this example here. You can see this gene AD15, it shows very low count data across all samples, whereas you have this gene here AD2, you can see very high expression, right, very high count, okay, meaning this gene probably is more highly expressed. Of course, this is raw count data, so we have to be careful in connecting these to the expression levels, because we have to do some normalization before we can do that, okay. So, I will show you, right. So, as I mentioned, DSEC2 uses this median of ratios method, right. So, it actually calculates something called the scaling factor or normalization factor, and this package is also called the size factors, right. So, in DSEC2 package you will see it will call as size factors. These are nothing but the normalization factors or scaling factors calculated in median of ratios method. Okay. And here are the steps. So, if you run DSEC2 on this sample, so here are the 5 steps, right, 6 steps actually. So, the first step is estimating size factors. So, this is actually you will see when we do the hands on, it will actually give you all these steps one after another. So, the first is estimating size factors. This is where the normalization is taking place, right, using this median of ratios method. In the next step, it is actually estimating dispersions. You understand now what is dispersions and how it calculates dispersion. These alpha i's, right. For each gene, you get this dispersion estimate. So, you have this gene wise dispersion estimate, right. So, you are getting this gene wise values, and then the next step is of course the dispersion shrinkage, right. So, we mentioned, right, why you need to do the shrinkage on dispersion. For doing that, it needs this information on mean dispersion relationship, right. So, you need to generate this something like mean expression or mean count versus dispersion, right, and you will probably see something like this. So, you will see these points, right. If you see the original paper, you will see these points around, and this kind of relationship you will see. Okay. And for some genes, right, where you have like higher dispersion compared to these genes of similar mean expression, right. So, that is the assumption in this model, right. The genes with similar mean expression should show similar dispersion value, right. So, imagine one gene here that has higher dispersion. So, for that gene we need to perform the shrinkage, right. So, of course, the method will perform that shrinkage again depending on the number of replicates that are present in the data, right. So, it will bring it closer towards the expected value, of course, not to the exactly to that expected value, but closer to that expected value. So, by doing that, right, so it will generate this final dispersion estimates, right. So, this is what it means by final dispersion estimates, because it is doing this dispersion shrinkage, right. And then the final step is the fitting model and testing, right. So, fitting model is this generalized linear model, and the testing part is the hypothesis testing. So, this generalized linear model will give the estimates for these coefficients beta values, right, and then you have the hypothesis testing to identify genes, right, that are differentially expressed. So, it will simply generate the test statistics, the wild test statistic and the p value, right. And of course, it will also do a p value adjustment for multiple hypothesis testing. Okay. So, let us now look at the results, right. So, how these results actually look like and what you get. Okay. So, before we actually see the results, these are the actual size factors that were estimated by this tool DSEC2 on this data set. Okay. So, here you see for a number for each sample. So, for S1 this number is 1.127, so on. For S5 this is 1.10, right. So, these are the size factors that have been determined. So, you have to use this in the model, right. So, this will be used by DSEC2 in the model, right. You remember this count modeling, right, which is proportional to the fraction of molecules in the cDNA library and the multiplied by the size factor, right. So, Sj. Okay. So, these are the size factors for these samples. Now, in the results part, what we will get, we will see in a moment, we will get this mean expression of genes, we will get something called log to fold change, which is actually looking at the differential expression, right. How much is the difference in expression of a gene between two sets of samples, right. So, whether it is disease versus healthy or condition 1 versus condition 2. It will also give us standard error of LFC value. Okay. So, how much error is there in the estimation. It will give us the results of the statistical analysis. So, because DSEC2 performs world test, so it get world test statistic, you will get p value, and you will also get something called adjusted p value. This is a p value corrected for multiple hypothesis testing. So, you will get all these values for every gene. Okay. So, if you have 7,000 genes, you will get these values for 7,000 genes. Okay. And from that you can then identify which genes are showing significant difference, right, or significant difference in expression. Also you get the log to fold change. Okay. So, here is the actual data, right. So, summary data that we will get once you run DSEC2. Okay. So, it says log to fold change, and we have done this being G4 versus G1 analysis, right, because this makes sense. G1 and G4, they are quite different, and it says there are about 6,805 rows, means 6,805 genes and 6 columns here in the results. Okay. Because we have this base mean, so this is the average expression of the gene. You have log to fold change, you have standard error of log to fold change, you have statistic, that is the wild test statistic, you have the p value and the adjusted p value or p adj, right. So, you get these 6 columns in the results, and then you have the first column is for genes, and for every gene you can see these values, right. So, here is a mean expression for this gene ASC1, you have log to fold change, right, you have standard error, statistic, p value, etc. Now, you can see like this p value is quite significant, it seems, right. So, 0.0001, and you also get the p adjusted. Okay. And once we understand how p adjusted is generated, we can probably better interpret the data. One of the things you probably noticed, right. So, this log to fold change, right, it can be positive or negative, right. So, positive log to fold change means this gene is overexpressed in G4 sample. Okay. So, because this is a G4 versus G1 comparison, so G1 is used as the reference. So, this positive number means this gene AC1 is overexpressed in G4 sample. Okay. Whether this is significantly different, right, or significantly overexpressed, that will come from the p value, and we have to interpret the p adjusted later on. Okay. A negative number, right, minus 1.36 for ASC3 would mean that this gene is lowly expressed. So, it shows lower expression in G4 sample compared to G1. Okay. And what is the difference in log fold change is about 2 to the power 0.86 in case of ASC1, and in case of ASC3 is 2 to the power minus 1.36. Okay. Because we have to do that, because it is a log 2, right, log base 2. So, we have to take the 2 to the power. Okay. So, we have these results for all genes, and this is before LFC shrinkage. So, I have talked about this LFC shrinkage, right, why we need to do that, and this is results after LFC shrinkage. Okay. So, here the statistic part is gone, that because we usually do not use that statistics much, right. We usually are more interested in these p values, p value and p adjusted, and the log fold change, right. So, you can see these numbers have changed slightly because of this LFC shrinkage. For example, for AC1 it was 0.86 or something before this LFC shrinkage, here it is 0.78. Okay. So, these numbers will change little bit again depending on the number of replicates that are present in the data and its mean expression. Okay. So, we have now seen the results, and we know like what to expect after we have done the analysis, right, how it would be appearing. Now, we need to visualize it, of course, right, because we have these 7,000 genes, we cannot go to every individual gene and see its results, right. So, we want to plot the results in such a way that we can interpret or identify number of genes at least in the data set. Okay. Of course, there are different ways to do this in R package, in DSEC2 we can do that, in R we can do that, but also we want to look at the results, right, and with some plots, because visually we can then identify certain important things. Okay. And so, that is what we are going to do now. Okay. So, there are 3 different plots that we use very commonly. So, one is called MA plot, the second one is volcano plot, and the third one is heat map. Okay. So, I will discuss these different types of plots, and we will actually plot the results that we have just generated using these plots, and you will see they will give us quite informative inferences. Okay. So, the first one is MA plot. So, what we look at here is the mean expression versus log to fold change. Okay. So, how do you calculate this? In a moment I will talk about this. So, we will calculate 2 parameters M and A. So, what happens is that when you plot this mean expression versus log to fold change, you can actually see number of differentially expressed genes, okay, and their distribution of log to fold change. Okay. And also MA plot helps in proper normalization, right. So, we can check whether we have done proper normalization, and this was actually very useful for microarray data analysis, but here also in analytic data analysis we can say okay whether the normalization has been done properly or whether there is a need for another round of normalization or a different normalization. Okay, so into the technical details how we actually generate this plot. So imagine this scenario again, diseased versus healthy samples, okay, and for all genes I, we can say di is normalized count for gene I in diseased sample and hi is the normalized count for gene I in healthy sample, okay, and we calculate these two parameters m and a for each gene, okay. So mi is log 2 di divided by hi and ai is the log 2 di plus log 2 hi divided by 2. So a is kind of like the average expression, okay, and mi is the log full change, right. We are looking at difference in expression, okay. So here is the plot actually, how this is how it would look like. So x axis you have the mean of normalized counts or the a values, right. This is the average value, and then along the y axis you have the log 2 fold change or m, okay. And as you can see, the log 2 fold change can be positive or negative, and we have actually discussed this scenario, right. So positive change would mean the gene is overexpressed in G4 sample, negative change would mean this gene is underexpressed or show lower expression in G4 sample compared to G1 sample, okay. All right. So now what you see are these dots. So each dot is a gene, results for a gene. So we calculate this m and a for each gene and then we plot this, right, and this every point that you see is a gene, okay, and you can see these two different colors, right. So you have points in two different colors, some points are in gray and some points are in blue, okay. So what is the difference? So the gray points, these are not statistically significant, okay, and blue points, these are statistically significant, right. They show statistically significant difference in expression, okay. So this means these genes that are in blue and in this top part, these are the genes that are overexpressed or show higher expression in G4 sample compared to G1, and this gene blue in blue here, right, that show negative log 2 fold change, and in blue color they are showing lower expression in G4 sample compared to G1, okay. So you can kind of get an idea, okay, these are the genes, these are the number of genes that are showing this kind of thing, okay, and you can also see the distribution of log 2 fold change. So you can see if there are quite a few genes that are above these 2 value in log 2 fold change, right. So that means they are actually showing 4-fold overexpression, right, 2 to the power 2. So that is 4-fold overexpression in G4 sample compared to G1, and similarly you can interpret the data for the genes that show very low log 2 fold change or negative log 2 fold change, right, and you can understand they are showing significantly lower expression in G4 sample. Okay, so this is before LFC shrinkage, right. So what happens after LFC shrinkage is that you see, right, the genes that showed very low mean expression or mean count, they kind of disappear from this here, right. So one of the points probably notice, right. So we see most of these blue genes only in the high mean expression. For this gene that show low mean expression, right, even though you see the log 2 fold change is high here, right, they are not statistically significant, okay. This is because there is a lot of variation in the data, and because of this variation, when you do the hypothesis testing, it cannot identify, right, the p value is not significant, it cannot identify it as a statistically significant difference, okay, because the statistical test it depends on the variance of the data, right. So for genes that are that have low counts or show low expression, they show very high variability, and that is why they are not identified as significantly different, even though the average log fold change you can see is quite high, right, but even then they are statistically not significant. So what LFC shrinkage does is kind of shrink these LFC values, right, because they are highly variable, it actually uses this empirical based method to actually shrink these values, okay. And what you see is that these values have been shrink now, and you can see they are kind of closed around 0, right, and this kind of makes it actually very robust, this method, right, because we are kind of taking this variability into account depending on the number of replicates. Okay, so I also mentioned, right, we can check quality of normalization from MA plot, right. So this is something researchers have been doing for microarray data as well, and this can also be extended to RNA-seq data. So one of the assumptions in this kind of analysis is that most of the genes do not show any difference in expression across samples. Okay, and this means that most genes, right, their log to fold change value should be 0, right. So they should be lying on that 0 line. Okay. And if you see a dependence of log to fold change on mean expression, that would mean it probably requires better normalization, or maybe you need to use some other normalization on top of the current method. Okay. So what I'll mention right here, what you see is that most of the genes are lying on this 0 line. So this seems pretty good, but sometimes what you might see is that if you have the 0 line, the samples are lying like this. Okay, most of the samples are below 0, or you see some sort of curved nature of this plot. Okay, instead of this kind of nature centered around 0, you see maybe this plot is just slightly curved. Okay, now if this is the case, that would mean, right, this is the 0 line, right. If this is the case, this would mean you need better normalization methods. Okay, and this by inspecting this MA plot, you can probably say, okay, the data is properly normalized, or whether you need some sort of normalization or different normalization methods on this data. All right, so the other plots that are there, so another is the volcano plot. Okay, and this actually is looking at log to fold change versus log 10 p value. It kind of gives you an information about how log to fold change values vary with significance level, right. So this is what we get. So x axis is log to fold change, right, the LFC, and y axis is the minus log 10 p value, right. So higher minus log 10 p value means more significant, right, because you have the minus. So if you have, let's say 10 to the power minus 2 p value, your minus log 10 p value would be 2, right. If you have 10 to the power minus 10, your minus log 10 p value would be 10, right. So higher value of minus log 10 p value means more significant, right. And as you can see, we have set this threshold here, and based on this threshold, these genes are colored, these are significant, these are in blue. The genes that are not showing significant difference, right, there is no statistical significance, right, they are shown in gray here and in this part. You can also see like what fraction of genes are showing higher log fold change higher than 1, or genes that are showing log to fold change lower than minus 2 or minus 1, right. All those things you can visualize using this volcano plot. And finally, the heat map, right. So we use heat map for comparison of expression levels of a set of genes across many samples and not just 2 samples. And here is a heat map for the same thing, right. So you have G1, G2, G3, G4. So we are looking at, let's say, one replicate of each, and we are looking at the genes, different genes which are numbered here along the rows here, right. So you can see gene 1, gene 2, etc. So here you are seeing these expression levels, these are color coded, and the colors are given here. Okay. So if you have red color, this means this gene is overexpressed. Okay, it shows higher expression or positive log fold change. Okay, so you can probably see this very clearly here, right. For example, this set of genes are more highly expressed in the group 1 samples, right, compared to group 4 or group 3. In addition, what you can probably see is that there is kind of like a gradient, right. So G1 has the highest expression for these genes, then you have G2, they show some expression, and G3 slightly lower, and G4 almost no expression, right. So this kind of patterns you can identify with this heat map analysis. You can also do hierarchical clustering on top of this. So this heat map is without clustering, the genes are ordered, right, from gene 1 to gene 24, but you can do an hierarchical clustering. So it will cluster genes that show similar expression patterns across the data, right. So you can see it has clustered these group of genes because they show similar pattern across these groups 1, 2, 3, 4. Okay. So this set of genes, they have highest expression in group 1, then slightly lower expression in group 2, then lower group 3, and lowest in group 4. Okay. So we have talked about this difference visualizations that help us understand the results better. Okay. Here are the references for this class. So we have completed the differential expression analysis with the package DSEC2. At least I have discussed the theoretical steps. Of course, we will do it hands on, and we have seen the results that we get, and two results are very important for us. One is log to fold change, and there is p adjusted. We have visualized the results through MA plot, volcano plot, and heat map. Again, we have seen there are some important insights that we get from this visualization. Thank you.